Hello, in this video we will discuss about the node-like receptor. It is cytosolic receptor basically and form inflammasome. And this inflammasome character differently we will discuss. And total 23 node-like receptor divided into two categories. Node-like receptor P and node-like receptor C. A node-like receptor basically is the receptor a pyrene domain containing node-like receptor P, and C is the caspase, so the caspase containing domain. And let's begin to understand. First of all, I want to give the overview of their introduction and what is that and why node-like receptor is important. So, and in human, basically Alzheimer's disease, atherosclerosis, and gout disease is due to the node-like receptor P3 activation, and activation will rupture the immune cell to produce the substance which that form a plaque. For example, if we will discuss about the arteriosclerosis, which that is the deposition of the low dense cholesterol is due to the hypercholesteremia in blood is the LDL. So here is the blood vessel contain a LDL, low density lipoprotein, which that is due to the endothelial cell permeability will increase the LDL deposition into the tunica media. So here is the cholesterol crystal formation and atherosclerotic plaque formation, and this plaque will be increase and rupture and die the cell is known as the pyroptosis. So this is the pyroptosis is different of the apoptosis. So let's begin. Here is the cross section of the blood vessel is showing in the inflammation due to the recruitment of the LDL macrophage, monocyte and macrophage will deposit and engulf the lipids, and this lipid deposit and ultimately thrombus formation and rupturing of the endothelial wall to thrombus formation due to the blood clotting, and the heart attack and other disease can be occur. So in this way, here is the macrophage, which that is engulf the basically the lipids and cholesterol in the form of vesicle, and this vesicle will be hydrolyzed, and that is deposit in the foamy cell formation of the macrophage. This foam cell formation will activate the inactive node-like receptor P3, and this activation to become an inflammasome. Inflammasome structure is the node-like receptor P3 cluster formation, and this P3 cluster formation, node-like receptor P3 cluster formation, will be broken down by the ASC spec, and this ASC spec protein will be uh, cut this inflammasome from the polymerization in few minutes will be occur, and in this way this is the ASC spec will cut the part of the active caspase 1 from the pro caspase 1, and this pro caspase 1 will be activated and to become a cell death is known as the pyroptosis, and this pyroptosis will be occur. So we will discuss with the whole detail. This is the overview for understanding the disease relation with the node-like receptor. And these node-like receptors are present in the cytoplasm, not present in the plasma membrane, but present in the cytoplasm. So it is known as the cytosolic node-like receptor. So here is the active caspase 1 will activate the pro-interleukin 1 beta will hydrolyze or cut and to become a mature interleukin 1 beta, and this mature interleukin 1 beta is the basically recruit the lymphocyte others as well as other things which that is cause the inflammatory response, and it is the inflammatory mediators. So here is the macrophage which contain a toll-like receptor, and there are many antigen presenting uh, domain. So this is the macrophage. So let's begin to understand the plasma membrane of the macrophage for understanding the node light receptor. So this is the plasma membrane. This plasma membrane is look like this. This plasma membrane is bilayer of phospholipid. Contained inside is the hydrophobic, while the outside is the hydrophilic. And in this way, basically is the some inflammation or some pathogenic invasion will be caused the different type. So it means the hypercholesteremia is not the basic formation for the plaque formation, but also the pathogenicity will be occur, and that cause the different type of basically due to the inflammation of endothelial cell, and the tunica media will. So here is the different type of node light receptor are present in the cytoplasm. For example, here I am drawing the node light receptor P1, C4 and P3. So this is contain as a trimeric unit. In this way, you can see here is the leucine rich domain or repeat in leucine rich repeat, and here is the NaCHT domain, and here is the um, uh, pyrene domain, this pyrene domain. And here is the node light receptor P3 we will discuss. It is the inactive. It is required for the activation by the adapter protein ASC. ASC. It is adapter protein contain card and pyrene domain. This will be bound with the node light receptor 3, which that is the two subunit of the node light receptor P3, will bind with the pyrene domain, and binding with the pyrene domain, pyrene to pyrene with the, and here is you can see the card and pyrene is the adapter protein will bind and will be activate, and this activation will become an inflammasome when the procaspase 1 will bind with the card side. So this card side will be bind with the red color is showing in the figure. So after this card side bind, so the procaspase 1 is activated by inflammasome. So the procaspase 1 is activated by inflammasome, and in this way inflammasome basically here you can see this is the procaspase 1 will be activated to into the caspase 1, and in this way you can see in the figure. So this caspase 1 will be formed, and this caspase 1 is used for the cutting we will discuss, and in this way the basically the restriction type enzyme caspase 1, this caspase 1 is looking like this here you can see, and in this way here is the pro interleukin beta and pro interleukin mature pro interleukin beta, which that is cutting from here, and you can see that activation of due to the caspase 1, and in this way the caspase 1 will be activated, and that cause the caspase 1 here as you can see, and in this way the caspase 1 activation to cut the pro interleukin 1 beta to mature interleukin 1 beta. So here you can see here is the red color, you will not understand, you can see, and this basically is not here. So in this way you can see here is basically the pro interleukin 1 beta is caspase 1 activation to cut, and this mature interleukin 1 beta will be formed, and this caspase 1 activation is basically attached with the procaspase 1, it's basically with the inflammasome, and in this way activation due to the inflammasome, and inflammasome is important for the caspase 1 activation for the maturation of the interleukin 1 beta, and this is the inflammatory mediator will recruit the other lymphocyte is a cytokines release, it is cytokines, and will release, and here is the type 3 and type 4 bacteria secretion basically is the type 4 and type 3 channel, which that is basically the pathogen associated molecular pattern and death associated molecular pattern will be enter and stimulate the node light receptor P3, and it will be activated by this mechanism. So important thing is that for you. So here is the pyroptosis will be occur. This is the pro-inflammatory cell death. In this way, the pro-inflammatory cell death is the pyroptosis is due to the stimulation of water incoming through a membrane rupture, and in this way the rupturing of the membrane will be permeable the water movement inside, and the cell will be swell due to the excess of the water inside the cytoplasm, and in this way swelling will lead to rupture and death, and this death type is known as the pyroptosis. Pyroptosis, because it is the basically the pyrene domain is used for it, and it is the pyroptosis is a different mechanism. So the other apoptosis is the another way, which that is the inhibitor of mTOR pathway and etc., which that is the different and inhibit the growth factor. So that is apoptosis is the different, but here is a pyroptosis. So on the other hand, here is the plasma membrane will recognize the double-stranded RNA, double-stranded DNA, and CpG DNA of the pathogen will lead to trigger the inflammasome formation by the activation of node-like receptor P3. On the other hand, the ATP, excess of ATP and bacterial toxin will trigger the potassium efflux, which that trigger the activation of the inflammasome due to the node-like receptor P3 inactivation to become activate. On the other hand, the death associated molecular pattern and the pathogen associated molecular pattern will also trigger by the endocytose the lysosome, and this lysosome will be ruptured and produce the inflammatory mediator and the adapter and the reactive oxygen species and cathepsin B protein, which that will be activated by this mechanism, and in this way the cell will be die. So that can be thrombus formation and rupturing of the macrophage due to the foamy cell and crystallization formation and pyroptosis.